So in a previous section, we discussed a little bit about collision theory, even though we didn't, actually call it that. But we discussed how molecules, particles, atoms, whatever they are, have to collide in order to react with each other. And the actual postulates of this theory are that the rate of the reaction is proportional to the rate of the collisions. Our molecules must be oriented properly whenever they collide in order for the reaction to occur. And the molecules must have adequate kinetic energy to react. So this kinetic energy is what fuels breaking the chemical bonds. And then molecules with kinetic energy is too small, just will bounce off each other, but they won't have enough energy, to actually break the bonds and react and then form more bonds. So this energy that's required for the reaction to happen is called the activation energy. So the activation energy is the minimum energy necessary in order for the product to be formed. So you can think of activation energy like whenever somebody is golfing and there's a little hill that they have to get the golf ball over in order for it to reach the goal. This little hill is the activation energy. Reaching the goal is the product. So here we have a reaction energy diagram where we start off with our reactants and our products are over here. So this is just the reaction occurring from reactants to products. The y-axis is the energy necessary for the reaction or given off. So we start off at a certain, value of energy. In order for the reaction to occur, there has to be some energy input, and that's the activation energy, okay? So this difference right here between where the reactant started and that peak is the activation energy. So you'll get something that's an intermediate that you can call an activated complex. The state of the system at the activated complex is called the transition state. So that's at the very peak. And then from there, your energy will decrease and you'll get a difference in energy when your products form. So far, in Chem 1, you probably have the idea that chemical reactions just occur. You start off with your reactants, whatever they may be, you end up with your products, right? But what actually happens is that reactions, they can occur like that all at once, where you start off with your reactants, they react, you get your products. But they can also happen through several steps. So each of these steps is known as your elementary reaction or process, whichever way you want to call it. And we have a term to describe these elementary reactions called the molecularity. So molecularity has to do with what is involved and how many molecules are involved in that step of the mechanism. So, for example, when you have a reaction that simply goes from A to your product, so a single reactant to your products, we will term this unimolecular. Remember, because the rate law is only affected by that one reactant. When you have bimolecular reactions, you will have two reactants affecting the overall rate law and how fast it happens. And then it would be three, whichever combination of these three, and then this is showing you the different possible rate laws. So also notice how you can have just one reactant affect the overall rate law, and whenever that happens, the order goes up. So for example, here, the order with respect to the concentration of A is second. Here, it's third. Here you have second order A, first order B, and so on. So how does having multiple steps affect the overall rate laws? So the overall reaction cannot occur faster than the slowest reaction of the mechanism. That sounds kind of weird, like a riddle or something, but I want you to look at this picture, and I'll zoom in. Here we have a tollway, and we have two toll plazas. Here in this first situation, the cars are slowed at this toll plaza, and the cars are going pretty fast through that second plaza B. On the second situation, we have car is going pretty fast through A, and then B is what's stopping, slowing the traffic. So both of these, their overall rates are affected by their slowest rates. So here in situation A, or first situation, the rate determining step is through passage A. That's the one that's, taking long overall. It's affecting how fast you get home or whatever it is. For the second situation, your rate determining step is through passage B. So this is the one that's overall affecting, how fast you get to where you need to. So again, most reactions don't happen in one singular step. You may have 2 or even 3 steps that make up an overall mechanism. So let's take a look at a mechanism with a slow initial step. So overall, this is our reaction that's occurring. Now there's 2 steps to it, but overall this is what we start with these reactants and these products right here. So in this type of mechanism, you'll have a first step and then a second step. And in this particular example, this step is slow and this step is fast. Whenever you have a reaction like this with a slow first step, the coefficients of the reactants will be the same as the order in the rate law. So here we have 2 reactants. Each of them have a coefficient of 1, right? The NO2 has 1 and then the CO has 1. When you add this up, you get 2. So overall, this is our order. Whenever you have a reaction like this with a slow first step, the total coefficients on the reactants of the overall equation will equal the rate order. So that's why the rate law is that right there. So here we have the first step of the mechanism. Notice that we have something that is not present at the very end in our products. So what this is, right, the NO3 is not in the overall stoichiometry, so what this is an intermediate. So here I'm showing you graphically how we start off with our reactants. We have a transition state from our first step, we have an intermediate, and then we have another transition step with our second step. Now this is not showing you time, right? This is showing you the reaction progressing and the energy it took. So don't correlate this with time and anything happening faster or slower, because we know that the first one was slow and the second one was fast. So intermediates can occur, they can be stable, and you could even isolate or identify some intermediates in certain reactions. Now overall, when we add these 2 reactions up, right, 1 and 2, what you will end up getting is this overall equation. So we should end up getting CO gives us NO. So if you remember from Chem 1, maybe a little bit from Chem 1 about thermochemistry and specifically Hess's law, this is something you did. You added up several steps of a reaction, and we calculated overall enthalpy of the reaction. So this is something you kind of already did, but now we are working towards connecting it all together. And finally, we're going to talk a little bit more about catalysts. So catalysts, we briefly spoke about them and how they increase the rate of the reaction. So they make the reaction go faster. But how do they do this? So now that we've learned about activation energy, we can actually show an activation energy diagram and how a catalyzed reaction brings the activation energy down. So it makes the activation energy be smaller, whether it does that in, by breaking it down into steps, and in this case it does. So you can see here how catalysts can change the mechanism by which the process occurs. So in this case, you have 2 steps and you have an intermediate somewhere in between there. But overall, it made the activation energy lower in comparison to that uncatalyzed reaction. So in this example, we are going to use reaction diagrams to determine which of these diagrams show the reaction having a catalyst present. So remember that earlier we discussed that graphically we can calculate the activation energy by looking at the energy of where you started versus the transition state energy. So this difference right here is your activation energy, your EA. So we're going to calculate that for both of these graphs to determine which of them has a catalyst. Now, visually, you probably already are able to tell, but still, just to show you the difference in activation energy. So for A, to calculate EA, right, graph A, we would take the energy for the transition state, and that's about 32 kilojoules, minus the energy for the product, the reactant, sorry, and that's about 6 kilojoules. So you end up getting 26 kilojoules for your overall activation energy. Now for B, we start off again at 6, but our transition state energy is about 20. That line isn't the greatest. So we start off at 20 kilojoules, subtract the starting, which is 6, and you end up getting 14. So based off of these two calculations, we can see that the activation energy was lower for graph B, and this indicates that this had a catalyst present, which decreased the activation energy, which made the reaction go faster. So now we're going to quickly talk about some different types of catalysts. So we have homogeneous catalysts. These catalysts will be in the same phase as your reaction. So many times your reactants and the catalyst will be dissolved in the same solvent. And you can see here we have an NABR catalyst that's going to be added to the reaction mixture. We have aqueous hydrogen peroxide in acidic solution. So we add the catalyst, and then what you end up having is the reaction occurring, and you can see that by the bubbles forming. So here we have a different type of catalyst, and I realize now that I made a huge mistake. This should say heterogeneous, so I'll fix that on the slides. And for heterogeneous catalyst, the catalyst will be in a different phase than the reactant. So very often a technique of using this type of heterogeneous catalyst, you will have gases passed over a solid catalyst. So here our nickel is the catalyst. What we have is our hydrogen gas being passed over this solid, and what you have next is hydrogen adsorbing. So adsorbing is kind of like sticking to the surface of the nickel, and then from there you will actually have your reaction. So our reaction is our ethylene reacting with hydrogen. When this ethylene reacts with the hydrogen, you start off with a double bonded carbon compound. What you end up with when you react with the hydrogen is a single bonded carbon compound. So this technique is readily used whenever you are trying to produce saturated fats and oils. So you can start off with your unsaturated fats and oils, whatever those may be, and you complete this reaction with your nickel catalyst, and then the hydrogen reacting gives you saturated fats and oils. So this is definitely useful in the food industry. So they use these heterogeneous catalysts to convert those. So the last type of catalyst we have are enzymes. So enzymes are actually biological catalysts. They have regions where reactants attach to. We call this the active sites, and then the reactants are referred to as substrates. So there's just terms very specific to catalysts and enzymes specifically. So here we have catalase present in beef liver, and we are going to, so this is ground beef liver, which is kind of gross, but that catalase present converts H2O2 to water and O2. So normally this would take longer, the reaction of H2O2 being decomposed into water and O2 gas, but when you have catalase present, it happens much faster, and this happens to be an enzyme. It's a biological catalyst. So enzymes definitely make reactions go fast, especially in your body when your body needs to react right away. Here I just have a diagram of this, active site that we were discussing and the substrate. So substrate is your reactant. Active site is where the reactant, fits into in order for the enzyme to do its job and complete the reaction. So here we have the enzyme, and here we have the overall complex formed. So this is something called a lock and key model. The substrate fits very well like a lock and and key into the active site of the enzyme. Sometimes you will have something called an induced fit model. So this is the crazy thing about biology is, things can change depending on what you need. So enzymes can change the active site in order to fit to the substrate. So you see how it looks different, but then it ends up fitting. So there's a lot of different enzymes in your body, and they all do a bunch of different jobs. So what you have in your body is a bunch of different little machines doing a lot of work all at once, instantaneously. And there's different classes. You have oxoreductases, hydrolases, lyases, a bunch of different ones. Anytime you end it with aces, that's a enzyme. Each of them have their own functions. They all do different things, but they're all important in biological processes. And just to give you an idea of this particular family of enzymes called kinases, kinases do something called phosphorylation. So they add a phosphate group to proteins, to other biological molecules. But for example, let's say you are trying to mobilize glucose for that flight or fight response, right? Hopefully you've heard of that, but fight or flight is your body either determining like I have to run away or I'm going to fight whatever is about to like come near me or whatever it is. But the way it works is that you will have a stress signal, and there's a bunch of steps in between this. I'm just summarizing it really quick, but you will end up having epinephrine, which is adrenaline, bind to receptors on your cells. This causes a cascade of things, but what you end up doing is converting ATP. So you break down the ATP. That's where ATP comes into play. It provides the energy to get the reaction started. And you end up producing something called cyclic AMP. The first, or one of the major proteins that induces the rest of this cascade is protein kinase A, so kinase. So protein kinase A does a bunch of things, but just look at what it's doing. It's phosphorylating. That P is just a symbol for a phosphate, meaning it got phosphorylated. And overall, I know this looks really complicated, but overall what it does is it produces glucose to be transferred and mobilized out of the cell. This is a liver cell, so it's very special. It can do that. It can make glucose from scratch. And it also mobilizes glycogen. Glycogen is a storage molecule of glucose, so it's like a giant chain, this giant weird chain of glucose. So it breaks that down, and it overall gets a bunch of glucose ready to go out of your cells and to be transferred to wherever you need. So for example, muscle, it can be transferred over there now that your liver took care of that. And I know this like seems complicated. This isn't for you don't have to memorize any of this. This is just for you to understand. This reaction happens very, very quickly. So think about how whenever you suddenly get scared, your heart starts beating, and you freak out. That happens in an instant. And what's driving all of these things happening in your body overall is enzymes like kinases, and they catalyze these reactions super quick, like in seconds, milliseconds, I'm not exactly sure, but very, very fast.